So good evening, and this is chapter 11, chapter 10 from Bio 211 at Norwalk Community College. And what we're doing is we're looking at how cells, made skeletal muscle tissue, and we're now about to move into how do you actually get contractile proteins to function. And to do that, what I want to do is I want to review what we've done before, remind you that skeletal muscle tissue formed from myoblasts. Where did that go? Formed from myoblasts to create individual muscle cells, so these have all fused together. We have the individual muscle fibers that are made up of thin and thick filaments along with some elasticity and extensibility fibers inside there. But ultimately, we can break it all down to this really simple cartoon that has the sarcolemma that creates transverse tubules that allow electrical charge to pass across the surface of the membrane, adjacent to a sarcoplasmic reticulum that has a terminal cistern that has voltage-gated calcium channels, as well as active transport pumps that will help us move calcium back into that sarcoplasmic reticulum. But the true action hero, action heroine, action something or other, inside a cell is this thing that's called the sarcomere. And that sarcomere goes from Z disc to Z disc, and it's going to have thick filaments and thin filaments. And this is the contractile apparatus, regulated by the presence of calcium and the availability of ATP made from the mitochondria. We will then regulate how muscle cells function. I want to go here one more time before we get into all the details. If this is one sarcomere, notice it's Z disc to Z disc. You have an M line which goes through the middle. It holds all of these filaments in the correct orientation. So when you look down here, you can see myosin. Proteins make up the thick filament, and they have multiple heads that go in 360 degrees, and they interact with the actin filaments that are on either side of them. Okay? So when these begin to actually contract, what ends up happening is these globular heads literally pull on that corkscrew apparatus that looks like the thin filament. And as they pull, let me go to here. As they pull, remember, A pulls to B, B pulls to A, and we shorten the distance between 2 points. So that's actually what contraction is. Get out of that. So how does this work? The proteins themselves, they're inside muscle. There are contractile proteins, regulatory proteins, and structural proteins. The contractile will be the ones that are actually generating the force. Those will be the myosin proteins. Regulatory proteins switch the contraction process on and off. Troponin, tropomyosin. You could even argue the voltage-gated channel of the sarcoplasmic reticulum is a regulatory protein. Structural proteins, these align the thick and the thin filaments properly. The myosin line for the M line and the titin, okay? And actually also the dystrophin, because without the dystrophin, obviously, you don't get the conduction of force onto the surface of the cell, which would then not pull on the endomysium. So how does it work? Well, myosin itself, these are the thick filaments. It functions as a motor protein, literally a motor. Think of it as pulling on something. Remember when you played tug of war, you hand over hand, literally pulling on the string or the cord, right? It's going to convert ATP into energy of motion. So by cleaving a phosphate from ATP to give you ADP, you can actually pull on actin. And the actin are the thin filaments. There are a series of proteins that make up a polymer. They're regulated by this complex here, troponin and tropomyosin, okay? And in relaxed muscle, what happens is the troponin and tropomyosin block myosin interacting with actin. Okay? Calcium ions will bind to the troponin of the troponin-tropomyosin complex, and now it will allow it to move out of the way. That's why that calcium is stuck inside that sarcoplasmic reticulum. Once it gets out, it goes to the troponin. It now allows the troponin-tropomyosin complex to move out of the way, and now you can actually have movement by myosin pulling on actin. So that titin, I've pointed it out to you in a previous cartoon. stabilizes the position of myosin. Okay, it gives the elasticity and the extensibility, and then there's always that dystrophin. Okay, and it links the filaments to the sarcolemma itself. Having told you that, we can go through what's called the sliding filament theory, which is when the myosin heads bind to and walk, literally pull the thin filaments towards the M line. The Z discs come closer and closer together, and that's what's actually going to move all of this. Okay, so how does it actually happen? So let's go here. Here are the components, right? Here is our Z disc to Z disc, and there's that M line, and there's that H zone. And you can see it's all written out for you here inside table 10.1. But to get an understanding of how it works, take a look at this cartoon. Here are those myosin heads. Remember the myosin heads bind to actin? That's that actin piece there, but notice the troponin tropomyosin complex is in the way. Not only that, these have to be aligned so that they actually get into that groove and actually pull on it. So if you think about a bouquet of flowers, if you looked right down the end of this, you'd see that as they pull, they're going to change their location while they're pulling. So how do you do this? Well, you actually have to hydrolyze ATP at the myosin head. Textbook says energizes the myosin head. It changes the shape of it is what it does. So I suppose that does energize it, right? That's going to form a cross bridge. That's where myosin actually comes in contact now with the actin of the thin filament. And what's going to happen is because it's in contact and it's been potentiated by that ATP, it's now going to begin to pull on the actin. It actually pulls itself off of the actin, and what happens is it then snaps back and then binds to another ATP molecule. You'll see how this happens in a moment. Okay? And as long as calcium is there, as long as calcium is available, you're going to see that muscle contraction, okay, continues to happen. You'll see this here. Okay, so before muscle contraction occurs, calcium has been made available. That is the purple dot sitting to troponin tropomyosin, which means they've slid out of the way. Myosin cleaves the phosphate moves away, ADP and myosin bind to the actin. As it pulls, it releases the ADP and then snaps itself off of the actin, where it can then bind ATP again, and this continues through this sort of four-step mechanism as long as calcium is available. Okay? It also means as long as ATP is available. So if there's no ATP, this doesn't happen. If there's no calcium, this doesn't happen. That's why I would normally say that ATP and calcium are limiting to this. Okay? So how does it actually happen? Well, we have to couple it to excitation. So to get calcium available, what we actually have to do is we have to cause an action potential. You'll see in a moment what I mean by that. But every time I say action potential inside this class, you guys know there's going to be some event that will require some neurotransmitter, right? So, whoa. What just happened there? Let's get out of that. What you call hitting the wrong button in the middle of a presentation. Okay, so action potentials are going to cause the calcium to be released from the sarcoplasmic reticulum. So let's take a look at that. This is at rest. And what they should have done, of course, let's draw that in. They will at the end of the chapter. I don't know why they wait so long to do this. They're going to create a neuromuscular junction. And that neuromuscular junction will release acetylcholine to bind the acetylcholine receptor. And as long as your muscle is relaxed, these calcium channels here, active transport pumps, are trapping calcium inside the actual sarcoplasmic reticulum. So this would be no neurotransmitter. Over here, let's put this in. Let's put that in there. Let's release ACH to bind an ACH receptor, which causes now an action potential. And that's what those little red arrows are. But I'm going to draw my own little action potentials here for you guys. And as they travel down here, the calcium release channels, which are voltage-gated channels, will now release calcium so it can go decorate the troponin of the troponin tropomyosin complex. Notice no purple dots here, purple dots here. That's what allows for the actual coordination. Your textbook calls it excitation, contracting, coupling, but it's coordination of electrical event to now mechanical event. Once you understand it, though, so let's get rid of the cartoon. Get rid of that. Move to the next slide. there's an efficiency where the forcefulness of muscle contraction depends on the length of the sarcomeres. So when a muscle is stretched, there is less overlap. When it's shortened, there may be too little of an overlap. And why do we care about that? We care about that because every time you go to the gym, there's a sort of sweet range that says, look, this is the appropriate ratio of myosin heads to actin that can actually be pulled on. This is understretched. This is overstretched. This is your arm being too far away from the weight itself, and you've literally pulled the weight. So imagine you've 100-pound weight at the end of your fingers and you try and lift it up. Not going to happen. Okay, so that's what you're actually looking at here. I don't want you to memorize the number. I don't want you to memorize that number. What I want you to see is there is a ratio that makes the joint and muscle insertion and origin construction optimal. So how do we actually regulate this? Well, you know what we have to build. We have to build a neuromuscular junction. That neuromuscular junction is going to be the lower motor neuron that will form a synapse, okay, to allow for the neurotransmitter, and the neurotransmitter is going to be acetylcholine. So go like this. I'm going to go ACH, ACH. And that acetylcholine will bind to the acetylcholine receptors, which are going to cause now that action potential to occur. So how do you get release of acetylcholine? Well, if we're thinking voluntary muscle, think about picking your nose. You got to lift your arm up, stick your finger inside your thing, and squirrel away or inside there, right? But what I want you to do now is just assume acetylcholine is going to be released. It's going to bind to those receptors, and here's where you need to start paying attention. Okay? You need to release sodium into the cell. That will cause the action potential, which will then travel to the sarcoplasmic reticulum, opening those voltage-gated channels so that you can now release calcium. How do you shut it off? Well, once you've shut it off, you have to break down acetylcholine, the extra acetylcholine. So you stop. Somebody walks into the room while you're picking your nose, right? You stop. To make sure you don't move anymore, you rapidly break down that acetylcholine using acetylcholinesterase. Okay? And you know what this looks like. You've seen it inside lab. There's the neuromuscular junction, synaptic end bulb. This is the lower motor neuron. You can see there are many synapses around here to regulate the activity. That's actually how you get muscles to move quickly. More of these, more quickly they move, fewer of them, the slower it moves. Okay, release the acetylcholine. It binds the acetylcholine receptor. Sodium is allowed in. You cause that action potential. We want to break this down. We have to release acetylcholinesterase into this space. So now, let's put the whole story together. Look, nerve impulse into the neuromuscular junction. Acetylcholine binds acetylcholine receptor. Travels into the transverse tubule, opens up those voltage-gated channels. Calcium now exits, binds to the troponin tropomyosin complex. Myosin heads undergo that power stroke cycle by cleaving ATP, grabbing onto now the actin, pulling, and keeps doing this as long as calcium and ATP are available. However, when we want to stop this, you need to activate these active calcium pumps that will store calcium inside here by binding to a protein called calsequestrin. See if I can spell this for you. Calsequestrin. S-E-Q. I'm trying to do this with my left hand. Sorry about that. U-E-S-T-R-I-N. So inside of here is this protein called calsequestrin. So as this pump is moving it in there, it actually traps it in there. And that's now troponin tropomyosin complex goes back to its original shape, and now muscle relaxes. No calcium, no muscle contraction. You can't have calcium unless neurotransmitter has been released to activate the acetylcholine receptors to let sodium in. So again, here's that. Let's get rid of the cartoon piece. Go to the acetylcholinesterase here. Okay, it removes the acetylcholine. Okay, it terminates. I was going to say what it would do is actually strengthen weak muscle contractions. We don't need to worry about that for your exam. So how then, okay, this would be a good time to stop, believe it or not, because I want to move then now into muscle metabolism. Okay, so just to review, let's do this. We have a muscle cell inside your body, and we're going to say this is skeletal muscle fiber, so it's got multiple nuclei. We'll put a multiple nuclei here, nucleus here, nucleus here. I should have drawn transverse tubules in for you. Transverse tubule, and you got filaments inside here traveling the whole way across. And remember, they're made up of Z disc to Z disc, and they have thick filaments. Well, these are actually the thin filaments first, right? Thin filaments first, and then thick filaments. We'll pull on them. And by having neuromuscular junction to release acetylcholine, to bind acetylcholine inside here. Oh, that's right. Okay, acetylcholine to bind the acetylcholine receptor. We cause an action potential. This is our sarcoplasmic reticulum with voltage-gated channels. We now release calcium. It's going to come over here and bind the troponin tropomyosin complex. And now, because there's ATP inside here, we have muscle contraction. When we want to stop that, that's when we use the acetylcholinesterase. I don't know why they call it anyway. Okay? To actually break this down. Okay? In fact, let's get rid of that circle there. Make sure you understand. What I'm actually talking about is acetylcholinesterase here, not anticholinesterases. Okay? Cool. It's a pretty good place to stop.